This part could be a little bit trickier for your third grader, and I'd like you to give them some assistance in thinking through and perhaps editing their procedures. It's a good idea if your student is going to write in pen to first write it in pencil and then go over it in perhaps felt pen. Either a medium tip or a fine tip felt pen works best. For this inquiry project here, up in the right hand corner you'll notice it says Fair Test Design. We spent a lot of time this year talking about what a fair science test is. That if you're comparing something like how quickly water will freeze between salt and fresh water, there should be only one difference in the experiment, in the inquiry. There should only be the one difference of the salinity. You'll see more about that later, but up in the upper right hand corner of this template is a place for the student to identify the one thing that is different in their inquiry. Here's the student's procedure. Number 1, run sink faucet on cold water for 2 minutes. 2, fill 2 1-liter bottles 2/3 full, cold water. 3, add 1/4 cup table salt to one of the bottles and label it salty. Number 4, shake both bottles for 2 minutes each with lids on. Number 5, fill 2 room temperature ice trays, one with fresh water, the other with salty water. Label the trays. One word that's missing in that procedure for step 5 is it should say 2 identical room temperature ice trays. Remember that the ice trays should be the same. Step 6. Set trays on same shelf of a fridge freezer, not deep freeze, but leave 2 inches of space between the 2 trays. Close door, start timer. Number 7. After 10 minutes, observe by gently jiggling each tray and looking at the top. Record the results. Number 8. Repeat step 7 until a full skin layer is formed on top of 1 or more cells. Number 9. Repeat steps 1 through 8 again for a second trial. Students are to run the procedure twice and keep data from both times they run the procedure. Next, the display should include a diagram. What makes a diagram is the fact that it's labeled. So here you can see there's a diagram of 2 1-liter bottles, one labeled fresh, one labeled salty. There's a picture that the water comes from the same faucet and the water should be pretty much the same temperature. Over here is a picture of the 1/4 cup of table salt, and it shows that that is added. And of course the procedure said that they are to shake the bottles for 2 minutes each. If they shake this bottle for 2 minutes to make the salt dissolve, they must also shake this bottle for 2 minutes. They have to treat everything the same except for the one thing, which is adding salt to this bottle. Next, this diagram shows that the ice trays are identical. There is an ice tray with fresh water and an ice tray with salty water. And one thing that I've shown here is how you can show a magnified version of something on a diagram. So here I've circled one cell from the freshwater ice tray, and I've shown a magnified, it even says magnified, version of that. And it's labeled, it says full skin layer of ice, meaning that there's a sheet of ice on the top beginning to form. And then there's a little explanation here. After 20 minutes, there was a full layer of ice across several cells of the ice tray with fresh water. So a picture really helps the reader understand and see what was done in the procedure. Also required in the project is a table of the results. You'll notice that our table must have a title. In this case, it says time, minutes, for ice to form. We have a column for minutes, an area for fresh, the data from the freshwater ice, an area for the data from the salty water ice, and then you'll see there's an area for data from trial 1, and this is hard for you to see, but this says trial 1 and trial 2 of fresh water. This says trial 1 and trial 2 of salty water. Down below we have a key, and the key tells us what the data means. The capital letter L stands for liquid in this case. S stands for a skin layer of ice, but only partial, meaning that ice is forming on the top of the ice cell, but it hasn't completely made a full layer of skin all the way across the top. And capital F, capital S stands for a full skin layer of ice formed on the top of the cell. We can look at this table and see that after 10 minutes in trial 1, the fresh water was still simply liquid. Remember, they jiggle the ice tray and observe it. In the salty water trial 1, it was also liquid. But then, 20 minutes in, the fresh water had a full skin of ice on the top of at least one of the cells. But when you look at the salty water for trial 1, it was still liquid. Now we look at the trial 2 data. For trial 2, in both samples, the fresh and salty water were still liquid after 10 minutes. But after 20 minutes, again, there was a full skin layer of ice across one cell of the ice tray. However, this time in the second trial, it's interesting to see that the salty water did have a partial skin layer of ice in one of the cells. It's important that the data is never made up. Whatever data is found, whatever results occur, they need to be documented, even if the inquiry project is inconclusive. Your child's template, which comes home, will not have the lines. It will have printed here trial 1, trial 2, trial 1, trial 2, and some horizontal lines. You'll need to add the rest of the lines for the chart. You may also use a graph if you know how to make bar graphs or pie graphs or whatever would be useful for the data. And last but not least, it's important to have the conclusion. In this case, the conclusion reads, both times, meaning trial 1 and trial 2, both times the investigation was run, a full layer of ice formed in the freshwater tray after 20 minutes, but not in the salty water And here's our conclusion. Notice it's underlined. Fresh water freezes faster than salt water. Now in this case, the conclusion the student had was the exact opposite of their prediction. So did they do a poor job on their science inquiry project? Far from it. The fact that their prediction was incorrect does not mean that they did not do a good job. It means that they ran an appropriate test and found the results. It's okay to disprove their prediction.